Hello everyone, this is Uma again from the Tasab Kore College of Pharmacy, Varnanagar, and now today we are going to discuss about the HPLC. The topic will cover up the introduction, mechanism, working, principle and the applications of the high performance liquid chromatography. HPLC is an abbreviation for the high performance liquid chromatography. Chromatography is a technique for the separation. Chromatogram is the result of a chromatography and chromatograph is the instrument used to conduct the chromatography. Among the various technologies developed for the chromatography, devices dedicated for the molecular separation called columns and high performance pumps for delivering the solvent at a stable flow rate are some of the key components of the chromatographs. As related technologies became more sophisticated, the system commonly refers to as the high performance liquid chromatography simply became referred to as LC. Nowadays, we can use ultra high performance liquid chromatography capable of high speed analysis has also become the more widespread. Only the compounds dissolved in the solvents can be analyzed with HPLC. HPLC separator compounds dissolved in a liquid sample and allows the qualitative and quantitative analysis of what components and how much of each component are contained in the sample. In the next, we will see the working and the different instruments used in the performance of the HPLC analysis. The solvent used to separate the components in a liquid sample for the HPLC analysis is called as a mobile phase and it is stored in a reservoir. The mobile phase is delivered to a separation column, otherwise known as the stationary phase, and then to the detector at a stable flow rate controlled by the solvent delivery pump. A certain amount of a sample is injected into the column and compounds contained in the sample are separated. The compounds separated in the column are detected with the help of the detector downstream of the column and each compound is now identified and quantified. In the instrumentation and the apparatus of the HPLC, we can see that going into the more details, HPLC consists of the variety of the components including the solvent delivering pump, degassing unit, a sample injector, a pre-column filter, a guard column, a column and detector, a back pressure regulator and the waste reservoir. As for the HPLC, the pump delivers the mobile phase at a controlled flow rate. Air can easily dissolve in the mobile phase under the standard atmospheric pressure in which we lag in. If the mobile phase contains the air bubbles and enters the delivery pump, troubles such as flow rate fluctuations and baseline noise drift may occur. The degassing unit helps to prevent these issues by removing the air bubbles in the mobile phases. After the dissolved air has been removed, the mobile phase is delivered to the column. The sample injector then introduces a standard solution or the sample solution into the mobile phase. Temperature fluctuations can affect the separation of the compounds in the column. The column is placed in the column oven to keep the temperature at a constant rate. Compounds selected from the column are detected by a detector which is placed downstream of the column. The workstation processes the signal from the detector to obtain a chromatogram to identify and quantify the compounds. Will cause the separation with the help of HPLC. HPLC can separate and detect each compound by the difference of each compound's speed through the column. There are two phases of HPLC, the mobile phase and the stationary phase. The mobile phase is the liquid that dissolves the target compound. The stationary phase is the part of the column that interacts with the target compound. In the column, the stronger the affinity between the component and the mobile phase, the faster the component moves through the column along with the mobile phase. On the other hand, the stronger the affinity with the stationary phase, the slower it moves through the column. The elution speed in the column depends on the affinity between the compounds and the stationary phase. Now we will see how we can read the chromatogram. The word chromatogram means a plot which is obtained via the chromatography. The chromatogram is a two-dimensional plot with a vertical axis showing the concentration in terms of the detector signal intensity and the horizontal axis representing the analysis column. Retention time is the time interval between the sample injection point and the apex of the peak. The required time for the non-retained compounds to go from the injector to the detector is called the dead time. The peak height is the vertical distance between a peak's apex and the baseline, and the peak area colored in a light blue is the area enclosed by the peak and the baseline. These results will be used for the qualitative and quantitative analysis of the sample component. This is the basic working and the principle of the high performance liquid chromatography. Now we will move forward for the standard operating procedures for the HPLC. First of all, we have to prepare a standard working solutions and mobile phase which has previously filtered through the syringe filter and sonicated for the 10 minutes. Then we have to switch on the instrument and we have to start the purging of the mobile phase to remove the air bubble in the tubing of the mobile phase. We have to allow the mobile phase to flow through the column for the saturation of the column. Now we have to open software, that is the Open Lab software on HPLC. Next, we will develop the method by setting method parameters in the instrument status, isopump and VWD and save the method. Next, we have to click on the file, open method control and download method. Then we have to start the baseline of the mobile phase and we have to correct it to the 0. After the completion of the auto 0, the inject analyte solution through the injection port after perking knob to the load position. Then we will start the HPLC run on the software and pull knob to inject position. HPLC run on the analyte will be observed as the chromatogram using the software and save it. After completion of the work, perform the column washing for the 30 minutes. Allow the mobile phase to purge for 5 minutes. Then we have to close the software afterwards, switch off HPLC instrument and the CPU. Thank you.